Hi, my name is Jonas Schulte-Schrepping and I'm a postdoc at the Systems Medicine Department at the German Center for Neurodegenerative Diseases, where I work on immune regulation in the context of chronic inflammatory diseases and neurodegenerative diseases using both single-cell omics technologies and bioinformatics. I'm also one of the first authors of the paper CD4 T cell calibration of antigen-presenting cells optimizes antiviral CD8 T cell responses. And in the next 2 minutes, I would like to tell you what this paper is all about. In our publication, we describe the sequential integration process of innate signals, such as type 1 interference and CD4 T-mediated T-cell help in antigen-presenting cells that optimizes their capacity to drive antiviral T-cell responses. What intrigued me the most about this work was the combination of reductionist ex vivo experiments and mouse models with large-scale single-cell transcriptomics data from COVID-19 cohorts. In a previous publication, we have described already the dysregulation of the myeloid compartment in severe COVID-19. And I was very excited to follow up on these findings and look a bit more detail into how these alterations in the myeloid cell compartment in the innate immune system affect the adaptive immune response. We uncovered that type 1 interferon-induced transcriptional adaptations calibrate antigen-presenting cells to rapidly activate certain transcriptional regulators such as NF-κB and FOS in response to CD4 T-mediated T-cell help. And this in turn then leads to the upregulation of a unique set of co-stimulatory molecules and soluble mediators that are critical for antiviral CD8 T-cell effector function. And this two-factor authentication process in antigen-presenting cells has also proven to be beneficial in HSV-1 infections in mice, but also in SARS-CoV-2 infections in human beings. I think my favorite figure in the manuscript is a box plot in the supplemental material. It is panel B of extended data figure 3. During the revisions of the manuscript, I continuously analyzed more and more single-cell transcriptomics data on COVID-19 samples that were published at the time. And I wanted to investigate how the genes that we found in our ex vivo culture experiments behave in COVID-19 patients with different disease severities. And as you might know, human data, clinical data can be very noisy and complicated due to the many factors that influence the data. So I was very happy to see how the signal became more and more robust with adding new samples that other people published and in the end get a very clear picture. Our findings contribute to the understanding of how innate and adaptive crosstalk regulates T-cell responses. And this is of course critical in vaccinations and infections. And of course we eventually hope that our findings will contribute to the development of new treatments and new vaccines.